biology, and welcome to a quick video going over the cell membrane. As we transition away from atoms and molecules into biomolecules and cells, it's important to remember that, again, structure and function are always really related, and that the organization of things at a really small scale, say atoms into molecules, has really large implications for scales of things at a bigger level, such as how cells interact with their environment. So, cells are the smallest living unit of life that we know of. They grow, they reproduce, they express genetic material, they use chemical energy, and most importantly, they regulate their internal self using a viscous fluid membrane known as the phospholipid bilayer, which we can see in this image here. It's two layers, hence the bilayer, and it's made up of these guys called phospholipids, which are an amphipathic molecule, meaning that they have a distinct polar and nonpolar region. The hydrophobic tails, which are fatty acids, tend to be built up of saturated or unsaturated fatty acids, and the top of the phosphate head is very polar due to the O, 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 and O atoms, oxygen atoms, bonded to a phosphate here. This means that the phospholipid heads are going to tend to want to interact with water-based solutions, either the blood or interstitial fluids, while the inner layer will want to deal with the cytoplasm, which is the fluid of cells, and then the two layers inside will be using their hydrophobic nonpolar properties to create a very large barrier inside. Now, this is really important when we consider fatty acids because depending on the type of fatty acid you have in your membrane, you might be more fluid as you have more unsaturated fatty acids, or you might be very viscous and thick if you have lots of saturated fatty acids. And this is because the straight, single, solid, saturated fatty acids take up far less space than the polyunsaturated fatty acids that tend to have lots of curves in their chains. This is important because fluid membranes with lots of unsaturated fatty acids tend to do better in colder temperatures because that spacing helps maintain fluidity when they tend to compress at lower temperatures. Things at lower temperatures tend to shrink and compress, while at the other hand, this has to maintain an internal versus external separation. So the saturated fatty acids are much better at warm temperatures because that spacing becomes less visible than if we had unsaturated fatty acids. More on that in a little bit when I demo what that might look like. Now, in terms of the phospholipid bilayer, one of the main functions of it is to regulate an internal environment separated from an external environment. And things are either going to get in or out of the cell through this membrane using a process known as diffusion. And diffusion is just the movement of molecules from one space to another. And this is primarily based off of concentrations. So if we have a lot of molecules outside of the cell, they're going to want to enter the cell based on differences in concentration. Secondly, smaller molecules are always going to move through a membrane faster and more successfully than larger ones. And lower polarity or nonpolar molecules will always diffuse faster as well, because these nonpolar fatty acid tails do not engage with or interact with polar molecules. So when this comes to fruition, in terms of either regulating neurons firing, regulating metabolism, having immune responses, controlling hormones, or more, different molecules are going to be able to get in or out of the membrane more or less successfully. And basically, we can break it down into 4 different categories. Oxygen, or any small nonpolar gas-based molecule, will be able to interact with the polar heads, and because of their nonpolar nature, will be able to diffuse through the fatty acids successfully. Sodium, which is small but polar because of that charge and something that's solid, will be able to get through the polar head, but they won't be able to engage with the fatty acid tails. The same thing is going to be true for larger solid structures that are polar, like glucose or other sugars. They're going to be able to engage with the phosphate heads based on that polarity, those positives and negatives, but not the fatty acid tails. And finally, larger things like proteins or hormones won't be able to get through the membrane as well. This is often why we see many protein channels or gates embedded within the membrane of cells, and that's going to be our next topic of conversation after we deal with membrane fluidity. Now, here's a good example of what a membrane looks like in terms of phospholipids. Notice we have two layers with fatty acids creating a hydrophobic region and phosphates forming the interior and exterior that is polar. Now we have 1, 2, 3, 4, 5 triangular molecules that can't right get through the membrane based off of spacing. Notice we have saturated and unsaturated fatty acids. Now, like the giant wavy-armed inflatable things that you often see at a car dealership, fatty acids aren't rigid in terms of the movement of their tails. They sort of are like a dog tail. They move back and forth, up and down, and the more double bonds they have, the more likely they are to change shape. So it wouldn't be without doubt to notice that over time, even within a second of a membrane, you might see fatty acid tails open and close, move and swivel, especially if they're unsaturated. This will make it easier at times for molecules to diffuse across the membrane. So that is why often a higher level of unsaturated fatty acids tend to have higher levels of permeability and diffusion based on these spaces filling. Now, these unsaturated fatty acids are good for maybe increasing permeability, but when we consider colder temperatures, they're also beneficial because they help reduce the amount of compression and compaction of the membrane. It has all of these saturated fatty acids here that compact quite readily because of the straight, not really flexible fatty acid tails, so we're removing any possibility of these molecules diffusing. Now, if we think about the other hand, when we have an increase in temperature, we notice that the fatty acid tails that are saturated don't space out as much as the unsaturated fatty acids. This makes it too easy for things to be able to become permeable, especially if they're things that we want to keep out or in of the cell. We want to keep sugars in the cell. We want to keep bacteria and viruses out of the cells. That increase in permeability can become too detrimental. So, if we want to review and revisit things we talked about, we know that our membranes are made up of phospholipids, which are amphipathic molecules, which have polar and nonpolar regions. We use this to help regulate the viscosity of the membrane and how fluid it is, which can have beneficial aspects in colder or warmer temperatures. This is important because this will help regulate how easily things get in and out of the cell membrane, because molecules are always going to want to move to a lower concentration, even if they don't belong in or out of the smell. Different types of molecules will pass through the membrane easily or not based on their polarity, size, and state of matter. Generally, gases, nonpolar, and small molecules will diffuse easier. And this is important when we consider the fact that membranes are fluid and fatty acids tend to move based on the number of single or double bonds in the structure. We're going to be using this in class today and in the next class thinking about how different things get in or out of the cell. But I hope this has been a good overview of the phospholipid bilayer, its structure, its function, and why it's important for cells.